Thanks very much for the opportunity to speak today. So Sernova is a publicly traded company. We are listed on the TSX up in Canada, on the OTCQB under SEOBF in the United States. And we have aspirations for NASDAQ at the right time. And we're also listed on the Zedra under PSH. And we are a clinical stage company. These are our forward-looking statements. So what is Sernova doing? So we are a cell therapeutic company, and we're looking at treating and finding a functional cure for lifelong conditions where patients are having to take injections of medications on a daily basis. And we have what we call our cell pouch system, and this provides the potential for a functional cure for these chronic diseases. The cell pouch itself is an implantable device, and we'll talk about that in a few minutes, and this is for the placement of immune-protected therapeutic cells. And what is really important about our device and devices in general is that if you want to have a device that can have therapeutic cells, you have to make sure that there's no ability to have a fibrotic reaction. And we've had devices in patients for multiple years without any problem. So our primary program is type 1 diabetes. And we also have additional programs for hypothyroidism and hemophilia A, which is an ex vivo gene therapy approach that we're taking. Currently, we are conducting a phase 2 trial at the University of Chicago, and we have patients that have been insulin independent on our therapies for over 2.5 years now. So we know that this product is moving forward in a successful way. We also have a partnership with Evotec, and this is for an unlimited supply of iPSC islet clusters that we'll talk about in a few minutes. So in terms of our capital structure, the bottom line is that we have about $45 million in cash and no debt. And this is giving us a runway for a number of significant achievements as we're going forward. So in terms of our integrated technology approach, we have developed what we call the cell pouch. And the idea here is that the device itself is a very, very porous device made into a series of chambers. And when we place this deep under the skin at the level of the muscle, we find that the body naturally fills in all of the pores of the chamber and forms these highly vascularized tissue chambers into which we can put the therapeutic cells. When the therapeutic cells go in there, they will naturally connect up to the vascular system, and they're sitting in a normal tissue matrix, just like regular cells do in terms of producing an organ-like environment. Our therapeutic cells, the typical approach that we've taken is to first work with human donor cells. This allows us to de-risk the program and test one variable at a time. And as we're moving forward, we are working on iPSC-derived technologies. And so when we talk about our diabetes program, we have already de-risked the program with human donor islets and are now moving into the iPSC technology clinical studies as we're going forward. Immune protection, our first approach is with the natural immune protection medications, and then we're also working on technologies around eliminating the need for these medications through a polymer approach, and then we're also looking at gene editing approaches. So in terms of our pipeline, you can see that our primary programs in type 1 diabetes, and then we're also advancing our programs for hemophilia A and for our thyroid disease towards the clinic. So with respect to our clinical program, our approach here is, first of all, to complete conduct of our phase 2 clinical trial, and by the end of this year, we're anticipating having the information to go to FDA in preparation for our phase 3 study. As we're going forward, our iPSC technology, working with Evotec, we're anticipating being in the clinic in 2024. So one of the questions you might answer is, why are we working with a cell therapy approach for type 1 diabetes? So if you think about over the last 100 years, patients have just been taking insulin injections, and the insulin injections can reduce the blood sugar levels, but they're not able to control blood sugar levels really tightly. And islets in a cell therapy approach has multiple cell types that can not only control the blood sugar levels and produce insulin, but they also produce a number of other hormones that can stabilize and raise the insulin levels and prevent the hypoglycemia effects from occurring. And insulin cannot do this on its own. So when we look at the size of the market, I think we all know that type 1 diabetes is a really serious problem. And the cost of diabetes is not insulin, it's actually the cost of all the side effects of the disease. In the cell therapy approach, if you provide the insulin, et cetera, and control blood sugar levels in a very tight way, then you can actually reduce some of the side effects that are occurring over a period of time and help these patients get back to normal. When we had a look at we did a payer study, and we discovered that in terms of being concerned about the side effects of the disease, payers were willing to pay between $200,000 and $400,000 per patient. And this gives you an idea of how important all of these hospital visits are in terms of trying to reduce the costs going forward. So in terms of our study design, we're working with patients that have severe hypoglycemia unawareness and long-term type 1 diabetes. And we're looking at, over a period of time, a number of, first of all, primary endpoint of safety, secondary endpoint of survival of endocrine tissue within our device. So we have small little mini devices that we remove after a period of approximately 90 days, and we do extensive histology to show that the islets are surviving and that they're highly vascularized. And then we're looking at a proportion of patients that have a reduction in hypoglycemia unawareness, also HbA1c levels, and then we have about 20 other additional endpoints that we're reviewing. So if you look at our first set of data that came out at the ADA back in 2022, what we found is that the first 3 patients that we work with became insulin independent, and the longest patient is at the 3-year time point at this point in time. So that's showing that we're not getting the fibrotic reaction, the islets are surviving long term, and the patient is doing really quite well. We did have 3 patients that did not maintain their immunosuppression drugs appropriately. We got those corrected and have, again, transplanted those patients again, and we're going to be providing data at the ADA meeting in June on all of the patients in the first cohort that we have. And we've been fortunate enough to be able to work with the FDA to have a second cohort of patients as we're moving forward with this trial. So to go back to the patient and how the patient is really viewing these things, our first patient that's been insulin independent for 3 years has taken over 20,000 injections, has been type 1 diabetic for almost 50 years, and they now are living a completely normal life. And this is what we would call a functional cure and what we're trying to achieve for all patients with type 1 diabetes. So in terms of our first cohort of patients, which are 6 patients, they have a very clear safety profile that we have been able to demonstrate with the device. No fibrotic reactions, cells are surviving, and we've been able to establish through different dosing of islets and dose density what we would consider to be the appropriate ideal dose of islets to be able to achieve significant efficacy. Our second cohort of patients, what we are doing there is providing an optimized dosing regimen in these patients, and we've manufactured devices that are slightly larger that can provide us about 50% more islets and also reduce the density of those islets so that they can have a lot more space for vascularization. We have already enrolled the first 3 patients. 2 of those patients have been transplanted, and we're anticipating the rest of the patients to be enrolled over the next number of months in 2023. Importantly, we've also engaged a new patient recruitment agency that is enabling us to recruit these patients into the study a lot faster, and this has been a real serious problem with a lot of the other companies in the field. So we've done really well from that perspective. We're anticipating having data on our 10-channel cell pouches in the second half of 2023, so look forward to reviewing some of the data that's coming out. And the results of that will be informing us on our phase 3 study that we are looking at going forward with, and still be working with the FDA on that. So in terms of our Evotec collaboration, which is really extremely important to the company right now, so we have an exclusive global license from Evotec to provide iPSC technology that has the ability to treat all type 1 patients moving forward. And the important thing here is that Evotec has put in a $27 million equity investment into the company, and we are working with them through the manufacturing process. Evotec is responsible for providing the GMP iPSC cells, not only for clinical trials, but also for commercialization purposes. And we're anticipating filing our first IND in Europe and North America in 2024. So this, we believe, will be the first iPSC trial for type 1 diabetes. So we're really excited about that moving forward. So if we talk a little bit about these cells, these are designed over the past 6 years to be fully formed islet clusters that have all of the cells that are required to be able to treat type 1 diabetes, and are designed to be very, very similar to human islets, produce the same amount of insulin, and are working exactly the same. They've also done long-term studies up to a year, showing that the control of blood sugar levels is very, very tight. And we will be presenting data in another couple weeks at the IPETA meeting in Boston, showing the first data that's coming out with the iPSC cells in our cell pouch. So we're quite excited about that. So we talk a lot about manufacturing process and how do you get a commercially viable product. So Sernova and Evotec have worked together to be able to achieve this so that we can treat millions of patients. And we need to have control over the product in space and time. And so what happens in the manufacturing process is that Evotec can get the cells manufactured up to a certain point. Those are able to be cryopreserved and held in space and time. And then when they are about to go to the patient, we can thaw those out, finish the differentiation, and then place them or transplant them into the patient. So this provides, I believe, the first opportunity for an iPSC treatment for diabetes to be able to be shipped anywhere around the world and be able to be used for millions of patients going forward. So in terms of our additional pipeline, as I mentioned before, we're working on a polymer approach that is more like a shrink wrap that can go around the cells and provide immune protection to those cells. And this is a porous cross-linked polymer material that has been shown to have no change in terms of the kinetics of insulin and glucose. And we have been able to show that these cells are surviving, producing insulin in our cell pouch chambers, and they're sitting right up against the blood vessels. So this seems like a very, very good approach to be able to eliminate the need for these anti-rejection medications going forward. We are also advancing our hypothyroid program into the clinic. And what we have shown is that we are able to take human thyroid tissue and place it into the cell pouch and allow those cells to be able to survive long-term and produce the hormones that are going into the bloodstream. And we've also conducted a long-term animal study now where we have placed thyroid tissue into the cell pouch in animals that don't have a thyroid gland, and we've been able to completely recover the feedback loop of the brain to the thyroid gland. So this program is going to be moving into the clinic, we anticipate in 2024. We also have an ex vivo gene-edited program in hemophilia A, and I'll just mention this very briefly, but this program has been shown to be able to take cells from the blood of patients that have hemophilia A. We've been able to insert the gene for factor VIII using a lentiviral approach and scale up those cells, place them in cell pouch, and we've been able to recover the function of the clotting in animal models of hemophilia A. And we're now in the process of optimizing this technology for potential moving into the clinic. So we have a series of important catalysts coming up. As I mentioned earlier, we're looking at the iPSC technology that's going to be released at the upcoming IPETA conference in Boston. We'll be having more information about our cellular immune protection technologies, and we're looking forward to the presentation at the ADA meeting, amongst a number of other activities. And then going forward in 2024, starting our clinical trial for the iPSC technology for diabetes. So thank you very much for the opportunity to present today.